think we're going to go ahead and get started. I'm Don Royal, the Chief of the Division of Aging and Geriatric Psychiatry here. And I just had a chance to talk with Dr. Rosano here for a while and got her off her schedule. So now I'm going to try to get her back on her schedule at the appropriate time here. So Dr. Rosano has an MD from the University of Palermo in Italy. She got a Master's of Public Health at Pittsburgh and has been doing cutting-edge imaging work in large epidemiological data sets. Now, there are a lot of large epidemiological studies out there, sometimes with thousands of people and longitudinal follow-up, but very few of these studies have significant biomarkers, and particularly neuroimaging, because it's so expensive and difficult to acquire in large samples. So she's doing some very innovative work by examining epidemiological data sets through the window of very cutting-edge structural and functional MRI and imaging. So her talk today is aging and motor control, and I'll let you, Dr. Palermo.
Thank you very much. Is this working all right for everybody?
Yes.
Okay, thank you. Thank you, Dr. Royal, for a great introduction. And I want to thank the organizers for having invited me. When I saw the schedule, I was a little intimidated by the fast pace that you wanted to subject me of all of these meetings. And then I realized what a privilege it is. I was reminded when you have these visits, what a privilege it is to just spend a day talking about science. And it's just fantastic. We spend a day doing a lot of other things, and oftentimes we forget about just brainstorming and talking about science. It was really a pleasure. And I also want to tell you how impressed I am with your students. I had lunch with some of them. They're extremely smart and brilliant, and postdocs as well. It's a testament, I'm sure, to the quality of the education of the center. So thank you very much. So I'll talk today about aging, central nervous system, and mobility. I'll talk about the aging brain, and I'll talk about mobility in older people. And I'll talk about how the aging brain and mobility are related with each other. I just came back from a ski resort in Colorado where I was extremely scared. And I was extremely scared about trying out the slopes for the first time. So I took a lesson in cross-country ski. I'm not very adventurous. And as I was learning with my teacher, she kept saying, watch me, watch me. So I wanted to try the things that she was telling me to do. But she was saying, no, first watch me. And that brought me back to one of my first rotation in rehab medicine, where that was exactly the same thing that the physical therapist was telling the patient. In that case was a patient with a stroke in the cerebellum. I have a very vivid image of the patient standing on the pillow, and the therapist say, think about your muscles. Think about your body. And again, you know, here is an invocation and an invitation to use your brain to move. That's what the instructor was telling me. Now, if any of you has ever practiced or is learning right now a new sport, I'm sure that you try to trace back the moment when you made a mistake or what you did that made you do it so well. Golf players, and I'm sure all sports, all athletes, watch themselves playing, watch other people playing. That is all cortical, right? And yet we think that when we move with our feet. So I'm hoping that with this little introduction, I've made you all convinced that the brain and mobility have something in common. And yet every time I started my talks, at least the very first few years, there was always someone who would say, well, how about the experiment? Who's not familiar with this picture? Anybody? Okay. There's a cat who's walking, even though the brain is no longer attached to the rest of the body. It is walking because there is some sort of a treadmill looking like a structure that makes the cat walking. The cat does not need a brain to walk, right? Geriatrician, how many geriatricians in the room? Okay. If a patient falls, I mean, sure, you may ask, were you paying attention? But the first thing you think about is the rest of the body, right? Joints, arthritis, pain, and so forth. You're not thinking, let me do a mini mental score on you. Let me see how your cognition is, let alone, let's do a brain MRI. Let's see what's going on inside your brain, right? And so we do not need the brain to walk like a cat walks on a treadmill, but we do need to walk with a purpose. We do need to walk to fulfill a certain plan when we go to a grocery shop and we're walking toward a certain place because we have a plan in mind, whatever it is that we have to buy. That is the type of a relationship that I'm thinking about when I say CNS and mobility. And everybody knows this song, right? I love it. I love the fact that it reminds me that there is something in the way we move that can tell us something about what's going on inside the brain. There was a very complicated paper published by Nakamura et al. Nearly 10 years ago that essentially that's exactly what he says. There is something in the way you move that can tell us something about the brain. In that case, the paper looked at patterns of gait and related it with mood and discovered that there was a strong relationship between mood that was tested using conventional paper and pencil tests with the patterns of gait. We also know where in the brain we should be looking for if we want to relate brain and mobility. This is a paper that was published also about 10 years ago, but the things that it's telling us have been known for a very long time. The prefrontal cortex is important for executive dysfunction, and which is important for gait disturbances. And then there is an entire circuit that involves subcortical structure that also affects gait disturbances through this integration of visual, vestibular, and proprioceptive sensory and contextual information into spatial maps. So there is substantial evidence that shows that there is an association between the central nervous system and the mobility. Most of the evidence, however, has been accumulated through studies of very localized pathologies. So focal diseases, say tumors or epilepsies, for example, epileptic crisis, animal models. When it comes to aging, again, it seems that just about everything is related to reduced mobility and maybe also the brain. But it's hard to identify where in the brain we should be looking for. So that's the reason why 4 years ago we applied for funding to begin examining these problems of aging, central nervous system, and mobility. And this was a 3-year cooperative funding initiative with the NIA. We had 3 conferences. And this is our conceptual model. So a lot of what I'll tell you today has to do with this conceptual model. So I'm going to walk you through that. And we'll start from the top, and then we'll go through all the way down to mobility decline, okay? So because of exposure to risk factors, which can include, of course, older chronological age, poor lifestyle, cardiometabolic and inflammatory diseases or conditions, we have abnormalities in the central nervous system that impairs mobility control. Now, when I say abnormalities in the central nervous system, I think about sensory motor brain networks, which directly affect mobility control, precentral cortex, postcentral cortex, cerebellum, and so forth. And I'm also thinking about other networks that are not strictly motor related. And they are visual spatial attention networks, information processing, and also memory brain networks. These networks all have to do with mobility control. They are all important in regulating mobility control, either directly or because they help the planning, the implementation of a plan, give salience and meaning to a certain motor plan. When this is impaired, there is mobility decline. Of course, you don't want to forget about all the peripheral contributors to mobility control. This is just a very simplified list. It's hard to include all of them. But basically, you have the peripheral nervous system, musculoskeletal system, cardiopulmonary system. I'm including here joints and all sorts of other non-CNS factors, okay? But that is the way we think about it when we think about the contributors to mobility decline. It is here organized in a semi-hierarchical way. In our conference, we gave greater relevance to the central nervous system networks because we felt that those are the networks that have been most overlooked. There is a lot of research on musculoskeletal control of mobility, muscle strength, for example, and mobility. And there was not, at the time when we started 4 years ago, we felt that that was the gap in knowledge that we needed to address. So the first year we met, we talked about this association here. What do we know about the association between changes in the central nervous system and mobility? What are the studies out there? We looked at animal studies, we looked at human studies. And second year, we looked at exposure to risk factors. and trying to understand. A lot is known about the association between all those factors that are listed there and the brain, but mostly in the context of a poor cognition, right? And memory decline, dementia, and so forth. Not much is really known in terms of the association between these risk factors and those networks that would be important for mobility. So again, that's what we did during the second year. And then the third year, which was just this past year, we tried to put it all together. We tried to understand what do we know in terms of ways to improve mobility in older adults, ways to delay the onset of mobility impairment, ways to slow down the mobility decline that occurs in older adults. What interventions are out there? My talk today will follow pretty much the same organization. The first paper was published. This is actually not a paper. Following the paper that was published, which is cited here, we also, together with Stephanie Studensky, which is the co-PI of this U13, we hosted a special issue on aging, CNS, and mobility. And we discovered, that was very helpful for us to see some of the most recent research in the field. And I invite you to look at that if you ever have the time. The special issue is very nice. There are just a few articles, but it's certainly worthwhile, glancing through that. So, of course, there is a lot of relevance in studying mobility impairment, and I'm sure all of you are familiar with the problems that I'm listing here. It's common and disabling in older adults who live in the community. 10 to 50% is the prevalence in some studies. It's 2-3 times higher than ADL disability. And the 20% which is in other studies, that would be the prevalence of neurological gait disorders. When I say mobility, a lot of what I'm thinking about is gait characteristics, and most of what I will describe today has to do with how fast individuals walk. I do have some findings that relate to other gait disturbances, say gait variability or unsteadiness, but mostly it's gait speed. I like to present this paper because the first time that I looked at this paper, I was looking at the height of each one of these bars. The prevalence of gait disturbances increases with age. Later, though, when I looked at this paper, I started looking at what was not in the graph. You can look at this another way. You can say among individuals 65 to 74 years old, about 15% have gait disturbances, but how about the other 85%? And then among 75, 84, there is a good portion also that does not have mobility gait disturbances. And even among individuals older than 85, you still have a portion of individuals who does not have gait disturbances. My talk today will try also to address this aspect of aging. There is an association between central nervous system and mobility. That can explain the reason why some people move worse than others, but I think that can also explain why some people move better than others. It can give us a sense of that capacity to compensate or adapt or resilience to aging that we often, think about and talk about, but we cannot quite quantify or identify what are the, the characteristics. Yes?
I know it's a detail, but how do you define gait disturbance? Because it has to be a continuous variable, and therefore to say it's either on or off is perhaps a little misleading.
Yes, well, this was a work done by Dave Bennett, and later also Joe Verghese did it. There are scales that you use to quantify gait characteristics. The GARS scale is one of them. And so it is a clinical appreciation of how a person walks. It's based on shuffling of their feet, irregularities, and how much they swing their arms. So it is ultimately a composite score of many things that you look at when a person is walking. This actually raises an important point that was so much the focus of all of our 3 conferences, and that is coming up with the unified measure of mobility, because we all have our favorite measure, of course, right? That's an extremely good point. And as in my discussions and conversations today, I was reminded of how important it is to also identify measures that are common across species. A lot of what I've heard today had to do with animal models and studies in mice, mainly. And oftentimes, I think when we're speaking this morning, we're talking about mobility. How do you measure mobility in animals that would relate to what we see in humans? It's an extremely good point. Thank you. So I became personally interested in mobility impairment because of its multifactorial nature. It has many, 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 many causes. I tried to highlight them before in the graph the diagram that I showed you, and we'll see that again. It is a highly variable process. The causes of this variability are not understood. And just to get this straight, this is what I mean when I say variability. Why do some people in this age group have gait disturbances and others don't? You can account for anything you want, and still you will not explain more than 50% of the variance of gait. And we know how hard it is, right, to explain a good portion of the variance of a certain health outcome. But as we have progressed over the past few years in our research, we have seen that physical activity is a possible intervention to reduce the incidence of mobility disability. There was a paper published in JAMA by the investigators of the LIFE intervention trial, where individuals who were walking 2 to 3 times a week, these were older adults, sedentary and somewhat frail, we saw that there was a reduction in the incidence of physical disability over 2 years. So that was very interesting and very important. But then the mechanisms are unknown. So we can modify it, at least for a certain period of time, but we don't know how or why. So most of my questions here have to do with whether we can explain part of this variability by looking at the central nervous system. These are other statistics of the prevalence of mobility disability increasing with age. And again, it's mostly for me to remind you that you always have individuals who will not develop physical disabilities. So here you have several statistics using different types of classifications of mobility, and they all point to the same thing. Yes, it increases with age, but not everybody are affected by it. And a lot of what I'll tell you, actually just about everything that I'll tell you, has to do with research that we have done in older adults living in the community. So we don't study patients with Alzheimer's disease, patients with Parkinson's disease. We study the common older adult. I think possibly the patient that really throws off the geriatrician because doesn't have any of those clear, right, things that would allow you to say, okay, this is exactly the problem you have. Okay, so back to this diagram. You can see that I have, again, identified these blocks here into 1, 2, and 3. This is how I'm going to divide my talk into 3 sections, and then there'll be a 4th section where I will entertain questions and discussion. But at the end of each one of these first sessions, I would like you to feel free to ask me questions so that we can move on. Do you have one?
So I saw in your slide just before you changed it, there were some numbers, and it gets back to the question that Dr. Nathaniels was raising. Could you just go a little bit more into, like, for example, in that figure on the right, what the mobility disability measurement was? I mean, did they have to reach a certain threshold of gait speed before they fell into that category, or what?
The EPC, no. In this case, I think the mobility disability was classified using a composite score of gait speed and the chair stance and holding the balance. So there are different scales that are used. A paper by Jack Guralnick, a lot before this was published by Suzanne Leville, a paper by Jack Guralnick and Luigi Ferrucci showed that out of these measures that you have in the short physical performance battery, or SPBB, they contain information that is also contained in the gait speed measure per se. And so they did very nice work to show that there's a strong association between gait speed and those other measures. And so that is also the reason why most of the study on mobility in individuals who are free from clinically overt neurological condition focus on gait speed. The paper that, the citation that showed up right before I changed the slide, just because I'm concerned about the time, is another important paper by Stephanie Studenski, published a few years ago in JAMA, that shows that there is a strong association between gait speed and mortality, a very strong association. And why is that? That's one of the things that we're trying to understand. There must be some shared pathophysiological processes between that, what determines walking more slowly and what determines the number of health-related conditions and then leads to mortality. Any other question? So there are two major emerging questions then. So this is just a different way of asking the questions that we have just been asking before. Why is the relationship of exposure to risk factor or to neuromuscular impairment with disability variable? Again, you may have different degrees of severity of impairment in any one of these systems, but not everybody will develop mobility disability. And likewise, why is the response to rehabilitative intervention different from individual to individual? You may have patients that, I mean, not two patients are ever the same, but you have patients with similar severity of pathologies, and yet one will respond much better to rehab than another. I mean, this is the variability, right, that every clinician encounters and every scientist encounters with whatever system we're working. So we think that the variance of mobility impairment in older adults may result from different degrees of brain plasticity that may buffer the disabling potential of other systems' impairment. So again, as I said before, I want to study the association between the networks in the brain with mobility decline, because I think changes and impairments here are contributing to mobility decline. But another reason why I want to study these associations is because I think that there is something here, inherent plasticity or capacity to adapt, that is going to help individuals develop mobility decline either later or more slowly over time.
This is a heretical suggestion. But at the beginning, you put up a picture of a cat that had cord cells, as you said, or something was disconnected.
The brain from the body.
So your prediction here is that you could put electrodes on someone and make an older person walk well again. If brain plasticity is the limiting factor.
Wouldn't be wonderful, but where do I put them?
Well, if the limitation is simply getting the signal down to the muscles, the joints, then theoretically, some of these people should respond by artificial stimulation.
Oh, yes, absolutely, right. I don't think that's so, I don't know if the question was heard, but you're saying, could the problem be that the signal is not going from the central nervous system down to the periphery correctly? It could also be that the signal goes down correctly, but is not being delivered, right? Either correctly or quickly enough. Maybe it's just a matter of speed, which I think it is, right? So the problem is that, again, we don't know where we should be doing this. Should we put these electrodes all over the brain? Should we put it in some areas? We don't really know, and that's precisely what we're going after. If spatial distribution of changes in the brain is specifically one of the very first things I did, because I wanted to understand where in the brain these changes would be, or these abnormalities would be, that would be related to mobility impairment. But you see, this is very much like the thinking process of a neurologist that wants to see where a certain change is so he can track it down to a clinical manifestation. But maybe that's not the case for aging, but we don't know, right? Or I would say, as you'll see in the next slide, I think we already know where we should look for. I think we have an idea of what networks are involved. We have known it for a while, and I think there is a good understanding of pretty much where we should be looking for these changes first. So the approach that I propose is that to understand the variability of mobility impairment in community-dwelling adults and to design interventions, we need to have cross-discipline approaches and also go beyond conventional disease-based models, because disease-based models, again, will call for a specific localized approach. Okay, so this is about halfway through the time I had allotted, right?
You have an extra half hour, right? Just an hour and a half.
Oh, but you don't want to be here for an hour and a half, do you? Maybe you should take a walk in between, though, just to make sure we are a little, okay. All right, well, here's the thing. This slide tells you everything that I want to tell you today, just about everything. So these are the first main 3 points that I wanted to make. First, lower integrity of the prefrontal subcortical and cerebellar networks is related to slower gait and higher step length variability, even after accounting for other factors. So that's a question that you asked right before. So if I were to put some electrodes, that's where I would be putting them. Or stimulators, right? So here we are in this section of my diagram. The relationship between integrity of these networks and mobility is explained by lower or slower information processing speed, indicating that these networks regulate both information processing and mobility. Now, this will be the second part of my talk and may be a little more complicated, but essentially it is saying that it is not only the association between the classical motor-related regions and mobility that matters. We also have to look at the association between parts of the brain that do other things, like information processing speed, attention, and memory. Because maybe the way the brain regulates mobility is because it increases the level of attention so that when I'm walking on a completely new terrain, I'm able to catch myself faster if there is some obstacle that I'm encountering, okay? And then lastly, a lot of what we have been doing in the very past few years has to do with this section here, and exposure to risk factors. We studied arterial stiffness, diabetes, physical activity, and systolic blood pressure. These are all related to lower brain structure integrity of these networks. They're also important risk factors for mobility disabilities, lower gait, and so forth in older adults. Okay, so we'll go through each one of these points one by one, and then we'll talk about the number 3, which is let's try to take it as all these factors together and see whether we have enough evidence to, not to guess, but to hypothesize what type of intervention would be most feasible to reduce mobility decline in older adults through intervening on the central nervous system. Okay, so we'll start with the first one, association between brain and mobility. Again, as I said, we're going to look at this. We kind of knew where we were supposed to be looking for, right? But when I started my work, actually, those are the MRIs that I had available. I had these MRIs available in almost, I think it was more than 3,000 people in a cardiovascular health study. It was an existing neuroepidemiological study. Actually, it started as a study of factors for stroke and other health-related events. And so all I had available was ventricular enlargement and white matter hyperintensities. So white matter hyperintensity is a measure of small vessel disease, among other things. And ventricular enlargement is a measure of brain atrophy, so how much the brain shrinks with age, essentially. And we found a very nice and linear association between these markers and gait speed. So this is the proportion of individuals with MRI signs. So having a ventricular grade greater than 5 meant that individuals had a very high severity of atrophy. It would be someone like this, essentially, a person like this, where this is more moderate, which would be corresponding to someone who would be probably, well, everybody here has a ventricular grade that is like that picture that I just showed you with severe findings. And those in the highest quartile of gait speed, which means, or lowest quartile of gait speed, which means for those who walk the slowest of all, were most likely to have higher atrophy as compared to those who were in the 4th quartile, which means they were walking at a gait speed greater than 1.2 meters per second. So next time you're on a treadmill, just check your gait speed. So if you're going at higher than 1.2 meters per second, that means you don't have a lot of ventricular atrophy or ventricular enlargement. The linear association was also found with white matter grade, which is a way to measure the white matter hyperintensity or small vessel disease in the brain. And then we found a similar association with brain infarcts. The associations were stronger for brain infarcts and white matter grade than for ventricular grade.
Excuse me.
Yes, please.
All of these people were without obvious cognitive impairment.
Thank you. Yes. These were voluntary. This is an epidemiological study of older age. To be enrolled in the study, you had to be 65 and older, free from cancer, free from dementia, free from disability. You were pretty well-functioning older adults. And these individuals they're still being followed, actually. It started in 1985, and they're still being followed. Yeah. So yes, to be in the study, you had to be free from clinically overt neurological conditions. We replicated the study in another neuroepidemiological study, this in Iceland, where they had another measure of brain integrity, which is called brain magnetization transfer imaging, in addition to white matter hyperintensities and brain atrophy. And we found very similar associations. It was a linear association the higher the burden of brain abnormalities quantified using these measures, the greater the probability of having a slow gait, even adjusting for all of these other factors. We also found that longitudinally there was an association between these neuroimaging characteristics and the probability of developing disability, and also the rate of decline in gait speed. In this case, we were a little more careful in selecting our participants. You see, we also want them to be free from stroke. This was a follow-up study, so it was done later. Free from dementia, free from disability, followed for another 5 years. So, but again, all we had was these so-called subclinical brain MRI characteristics, which are not particularly sensitive. They are not telling me where in the brain these changes are. The picture of Dr. Brack is here. She was also a Bison scholar, actually, and she helped me very much understanding all these measurements of gait using gait math so that they would be more precise. So later, we moved from those neuroimaging measures to more sophisticated measures of brain integrity, including volumetric measures. And those are objective so that we could address the variability that we found using the prior measurements based on rater. It's very subjective and scale. And neuroimaging methodologies that we use are automated so that we can examine large data set. And again, it allows us to look at localized abnormalities rather than global abnormalities. And this was the first time that we validated this measure. Dr. Eisenstein developed it in his lab. It's an automated measure that allows us to parcellate individual regions and get the gray matter volume. It was just a paper where we validated the method. So what did we find was now I was able to look at individual regions of interest. And this was the first paper where we looked at about 300 older adults, and we found that the regions highlighted here were the ones that were most strongly related to slow gait. So this is the prefrontal lobe and the cerebellum. So it was not a big surprise, right? Because more or less we would have expected that those regions would have come up, but the lateralization was very interesting. Mostly they were on the left side. And there is some theory as to why this would be that would have to do with some of those, the differential process of atrophy that affects the right versus the left hemisphere. We don't need to look at this. This is more to show you the relevance of the neuroimaging measures compared to age or compared to gender. In other words, for each unit of gray matter volume, each standard deviation counted and mattered almost as much as 1 year of age in telling us, in terms of the strength of the association with gait speed. We replicated these findings in another independent data set. This is derived from the Health ABC. This here, I want you to look at it just to appreciate the richness of the data set. These individuals were seen for the first time in 97, 99, and then in 2006 and 08, after in-person visits done in regular intervals, they received a brain MRI. And from that moment on, they received a neurological exam and muscle strength at each year, including measurements of gait speed and balance, cognition, inflammatory factors, and various health-related factors and conditions. And then 3 years later, they had the brain MRI, dementia battery administered. And then now we just completed the phone follow-up for disability. The reason why this is here is also to invite any one of you who has time to analyze data to please contact us. We have so much data that clearly we can't analyze it all. These are the two fantastic people who have helped replicate these findings. And sadly, by the time we replicated the findings, we didn't think it was important enough to publish, so we never did. But here is a slide that shows that we did. And the spatial distribution is very similar. It's, again, the prefrontal parietal. But we don't have much, here is the parietal. We don't have much in the cerebellum, but that is the problem with this particular protocol, where we were really focused on the frontal and parietal, and we kind of left out the cerebellum, except for a subset. Dr. Rosso, an assistant professor in our group, looked at another outcome, not gait speed, but step length variability. And again, she looked at regions, multiple regions in the brain, and she found that the strongest association was with the hippocampus and the anterior cingulate cortex. and the association was bilateral. So it was very interesting, right? This memory that we try to bring it into the game, in addition to anterior singular cortex, which is an indicator of executive control dysfunction, is very important for conflict resolution and so forth. So in the subset of individuals who had a good cerebellar measures, Dr. Nat Carney also studied the association between the volume of the cerebellum and gait speed, and also found a nice linear association. The smaller the cerebellum, the slower individuals were to walk. So here you have volume and gait speed. So this was the first section. Essentially, across different data sets, we looked at spatial distribution of abnormalities. We found prefrontal cortex, and we found cerebellum, but we also found some important subcortical structures, such as the cingulum and the hippocampus. Any question about that? Yes?
Especially one that's defects in the cerebellum, was there any other clinical parameters that we looked at, like proprioception, defects, and that nature, that certainly can be explained with sensory motor networks?
Yes, thank you. Thank you very much. So absolutely. We did a full neurological exam where we measured proprioception and we measured essentially all the sensory system. And we accounted for all those. We also have joint pain, which is collected through self-report. And we have muscle strength, which is an objective measure that we obtain using the King Kong chair. So it's a quad, so lower extremity. We have auditory, right? Information about auditory, right? A system. We have visual system. What other system am I forgetting? I think we counted pretty much. I mean, of course, these are not peripheral nervous system measurements done with the most recent technologies that were through neurological exam, but I think those are the ones that matter. I mean, they really capture certain subclinical, at least severity of signs. Did I answer your question? Yes, please.
There was some data you showed which suggested laterality. You said there was more effect on the left-hand side. Does this reflect whether they're right or left-handed? Is it always on the left side, or does that change if you're right or left-handed?
Yeah, so that was one of the very first things we looked at, but a very small number of people was left-handed in our sample, and we did control for handedness. So I don't think it has much to do with handedness, but I also don't think that we have enough numbers to distinguish that. The Harold model, anybody familiar with the Harold model of compensation, is also based on the evidence that one of the two hemispheres seems to be affected by aging faster, and the other one tends to compensate for the changes. And this would be the left and the right.
Was there any evidence of laterality in step length? Because older people tend to shuffle, and I haven't got any evidence for it, but it looks almost as if sometimes one step is longer than the other as part of the shuffle.
So it's a good question. We combine the left and the right, but before we combine that, we examine whether there is a difference between shuffling on the left, like you say, or difference between left and right. We don't find much. Remember, these individuals are free from neurological conditions. So we are really trying to capture them before any one of those things would be evident. So we didn't see lateralization differences in the left compared to the right. At least not that it would stand out, but I like that thinking because I think I understand where you're going.
Caterina, what about height?
Excuse me?
What about height?
What's the word you say?
What about height?
Cardiovascular system.
Actually, what I'm thinking about is my mum's partner is. I can, thank you. Yes, I asked, what about height, physical height? My mum's partner, for example, is 70 and a very tall man, and is now shuffling. And I think, I said to her last night, he's got a long way to fall. And it made me wonder whether tall people actually become psychologically much more prone to shuffling, simply because it's scary.
Because the center of pressure, the center of pressure is farther away, right, from the ground. So you're asking about height.
Yeah, is that a factor in the models?
Yeah. So we adjust for height. It would be it's a good anthropometric measure. It also accounts for the length of the legs, which would be directly related with how fast one person is walking. In our cohort, as I'm sure in many of the other aging cohorts, men tend to be taller than women because of greater advantages that received. And it's supposed to be dietary and so forth. But so adjusting for gender is also another way to account for that. In fact, adjusting for both gender and height by some is considered to be an over-adjustment, but we don't see an effect of that. Although what you're asking is a little different. You're asking fear of falling. And again, these individuals live in the community by themselves. We do have measures of falls, but that's not what we look at. There are some groups that examine this data set specifically for that, for that outcome. That would be interesting to look at those papers to see if they isolated height as a risk factor for fear of falling or falls.
Andrew was the author of that paper. Is she the one who did all the data?
What person?
I'm just curious about Andrea.
Andrea Russell?
Yeah.
So this analysis was a combined effort with Megan Olson-Hunt, who was a statistician, is a statistician. That time she was a student. And the modeling of the brain measures was a little more complicated than what we usually do. We use a PLS approach, a partial least square approach, actually sparse partial least square approach. So I liked it very much because it allowed us to do a lot more data mining than we typically do. We don't really know what area would be associated with step length variability, so we looked at a number of areas. And when you do this type of analysis, you really need to control for multiple comparisons and stuff like that.
I have a question about that. So in my very, very limited experience with MRI, I've seen that essentially the method and the algorithms that you use to actually process the data make a huge difference on the results.
Absolutely.
Yeah, so essentially by replicating it through, with different people, basically this is essentially talking or supporting the idea that this is
Absolutely.
fast enough.
Absolutely. The reason why, it's exactly the reason why we felt it was important to do it in another study. Now notice that what we did was, we did use initially voxel-wise based analysis where you do they're not really complicated, but essentially you compare this big three-dimensional matrices of data, which are essentially brain voxels between, say, a group with a condition, a group without a condition, and then you overlap these matrices and you find the areas where the differences are statistically significant. And that is where you can change the p and the multiple comparison, the alpha, and that things can change. To me, it was much better to show you how big this region is. It can be this many cubic centimeters in this group, and it is this many cubic centimeters in the other group. So a region of interest approach kind of made me feel that we could be more confident about what we were looking for. And then I can also measure the error in a way that I understand it, and I can explain it better. Yes?
So in terms of causality, do you say you see a similar relationship between these gait speed cerebellum in young cohorts?
I'm sorry, causality? What did you say?
Yeah, so I mean, you might predict that same relationship would be in younger individuals as well as older individuals. Have you looked at that relationship between cerebellum or white gray matter or any of the MRI
No.
measures?
No, we didn't. And I have to tell you, that I don't know that we would find the association strong enough in young people. The reason being that you don't have enough variability of the gait measures. And I think that the reason why our effect sizes in these studies are not very big is also because we're looking at individuals without overt neurological conditions. And so especially when you have them walking in a hallway where everything is the same for everybody, okay, and they're instructed to come with their sneakers on or comfortable shoes, they all receive the same instructions. I think we missed the opportunity to see a wider range of gait speed than if we were to test them out on the street while they're crossing, at the traffic light, and they need to figure out how fast they need to go. The shoes are different, maybe it's raining, and they don't know whether they're going to slip and fall, right? So I don't know that we would find these associations in young people. But I did not look. Anybody else? Yes. This is great. Lots of questions. I don't have to talk as much, although I really am. But
I
Well, what else could it be? If it's not, how else could it work out? It's not the brain, it's not brain change causing mobility, which is, by the way, not at all what I'm saying. I'm saying is an important contributor of gait changes, along with many other factors. You should not look at the brain in isolation, right?
Well, I guess, I mean, these factors could be changing throughout the brain, and you said you've looked at other brain regions and you do not see these correlations. So because you already know that these two regions are involved in regulating mobility, and you see a specific correlation there, that's one of your stronger arguments?
Well, so first of all, it makes sense. It has biological plausibility, right? The paper in 2007 that had that diagram with the brain and the networks, that has been shown in animals, disease models. So it has biological plausibility. Secondly, we find an associations between regions that I just listed here and gait speed, and do not find associations that survive multiple comparisons, or we don't find them at all between other networks and other regions. So that would be another, but the biological plausibility to me is the stronger argument. And the next set of evidence that I'm going to show you, I think it has to do with, it clarifies the mechanism of how this, why and how the brain, how is the brain really related to mobility, and I think it helps us discern a little more if there is a causal relationship or not. So why don't we look at those slides, and then, yes?
On your previous slide. Yeah. So just to clarify here, is the yellow are your regions of interest that you've drawn?
No. No. Yes, thank you. So these are examples from different methodologies. This is a result of a voxel-wise analysis. These are probabilistic maps. These are regions we drew.
Right. So the red then, is that indicating that the volume there is increased?
No, this is simply color. It's just a picture to show you where we find an association. It's nothing more than that. To find the strength of the association, the paper where that supports this finding shows the beta coefficient coming from linear regression models where the outcome was gait speed and the predictors, the main independent variables, were the gray matter volumes of individual regions. Yeah, so and this is actually yet another, I just like to look at brain pictures. So the other one is from functional MRI studies while individuals are doing a task of information processing. Yeah.
Caterina, it wouldn't really, give you a proof of causality, but are there any studies where interventions that are, supposed to engage a certain region, like, the people who are doing the unrelated, so we talked about that in the
At the big.
interest-
Yeah, yes, exactly.
So is there any data from that kind of intervention?
That's right. That's my last slide. We could skip everything else, but that's exactly what we need as a proof of concept, right? If you modify those regions through, for example, cognitive intervention, does your gait improve? And there is initial evidence coming from Joel Borghese in New York that there is some improvement in gait. It's transient. It's not for a long time, but I think it's pretty impressive. Yes.
That wasn't the point I was going to make, but I work in a field where human interventions are becoming more and more important. And even if you do an intervention and both change, they could both be secondary to something else.
Both
So
you're
even-
thinking CNS
Yes,
or cognition.
yeah. And a thing I like, which I realize may not be a major area of investigation, I like that last point on the slide there. I mean, as a physiologist, if I had to put a month's salary on this, I would say that the vascular effects are going to be fundamental. And so that's the point. If you did an intervention which improved the vascular system, then both your gait speed and these neurological, particularly the hippocampal stuff, would improve. And I think the data, one must collect the data, otherwise you'll get nowhere. I think the data are very fascinating, but this is a conundrum for all of these epidemiological studies. Even in the animal, it is difficult to design a study which gets a mechanism.
Well, but I think that's the only way to unravel the point, right? But you see, the brain here is on the causal pathway to mobility decline, right? There are risk factors. You're exposed to this, changes in the brain. But this very same risk factor are also going to affect all the systems. Again, I don't believe in one causal pathway leading from A, where A is one thing, to B, where B is one thing, to mobility decline. I don't think we can answer this question by looking at systems in isolation. I think the strength of, I mean, there are a lot of disadvantages, of course, in relying on epidemiological studies. They're descriptive, just for starters, but it allows us to measure all of these things in a large number of people. So you have enough variability of this measure, great range of values. So, but again, it's not to be found in one causing mobility decline. And that's not at all what I'm saying. It is it's one of the factors in the causal pathway, but it cannot be overlooked. And that's the reason why I think interventions to improve mobility have been so ineffective, because they're focused on one system at a time. Whereas by doing this way, we're missing the opportunity to do what we do in pharmacotherapies, where we potentiate the effect of one drug by combining it with another drug, for example, right? So that's exactly by targeting multi-system. I think the probability of improving this outcome is much larger. What are we doing here? We are doing that. Okay, so I'm going to try to explain why I think that one of the pathways linking changes in the brain, smaller volumes, for example, and mobility has to do with changes in attention and information processing speed. But if you believe it, we don't need to do this. Is there anybody? So would it make sense to you? The regions that I've shown you are related to information processing speed, okay? And so that would be that's one of the reasons why we start thinking about that executive control dysfunction could be mediating, explaining, at least in part, the association between the central nervous system abnormalities or impairments that we were seeing in gait. But prior to that, we had found a strong association between physical function and cognitive function. Much of the work that Dr. Royale has done here has actually inspired me to look into this association, executive control dysfunction and mobility. And they are strongly related, both cross-sectional and longitudinally. I'm putting up my papers here, but this has been shown by many, many, many other investigators in many different studies. That's the test that we have been using, the digit symbol substitution test, which is a test of information processing speed. Some say that it's a test of working memory, executive control dysfunction. It is a test also of hand motor agility. But it does a very good job in picking up reduction and slowing in information processing speed. And one of the very first things that we did was to see what parts of the brain were active when we were doing this, when individuals were doing this test. And this here on the left is, in fact, a probabilistic map that Dr. Venkatram did when he was a student with us, and now is a fellow at BLSA. And those are the areas that were most active. This is in older adults. When individuals were performing a computerized version of that task that I've just shown you, you have to match symbols and numbers. And then this is instead just to color the areas that indicate where we would find this association in another study, completely different study of individuals where we were also looking at executive control in the oldest old. So again, these are individuals older than 75 living in that community. So if you could keep in mind those, and that was one of the reasons why I had those pictures on the left when Don asked me what were they. It was to highlight the fact that we're always in areas of the brain that are not exactly identical, but they're very close to each other. And to me, that suggests that there are shared networks that are shared that overlap, and networks that are important for mobility and networks that are important for information processing speed. They are at least partially overlap, so that some of the structures are important for both information processing speed and walking, such that when you try to do two things at the same time, if that region is affected, you won't be as successful in doing it. So that's the whole dual task literature. I'm staying away from it, but that's one of the evidence, that provides the strongest evidence that these networks are in fact shared. And that might be the reason why one of the pathways leading from central nervous system impairment to gait might be through a reduced information processing speed. Now, there are 3 or 4 papers here where we've shown this different data set. First, I looked at the volume of the dorsolateral prefrontal cortex and the association with gait speed. And I found that when I adjusted for lower score on a test of information processing speed, the association went away. I looked at several other tests, and the digit symbol substitution test was really the one that had the strongest effect on in reducing this association. The other ones that showed up are also in tasks that are related to executive control function, and then one here, the finger tapping, that is possibly accounting primarily for that hand dexterity when you perform the digit symbol task. Dr. Nadkarni, I told you, had looked at the cerebellum, but he also looked at what cognitive tests might explain the association between the volume of the cerebellum and gait speed, and found the digit symbol, but not mini-mental score, explain the association between cerebellar subregion's volume with gait. And then another of our collaborator in Canada, Dr. Bolanzade, she looked at white matter hyperintensities and gait speed, and again found the digit symbol, explained the association between hyperintensities of tracts just in the frontal lobe, not anywhere else in the brain, and gait speed, but not mini-mental score. So those are 3 different papers that show that this could be the pathway that through which changes in the central nervous system, whereas volume or hyperintensities, primarily localized in the frontal lobes, are explaining the association between these changes in the brain and gait speed. So here we are at the third section, but before we get there, I want to know if there is any question about this point.
Caterina,
Yes.
I was curious about the prominent absence of the motor cortex.
The what?
Motor.
And what about it?
Motor pathways.
Motor pathways. So I'm thinking those would be primarily direct, right? They don't go through a specific cognitive domain.
Yeah.
So if I look at the volume of the presentral cortex, you mean?
No, when you were essentially going back to the first studies that you see that maybe at like with less prominence or less strong association? Did you see any evidence for motor like?
So the motor cortex,
Yeah,
you mean,
at the
right.
level,
Yes, we did find association between the sensory motor areas, right? We did. Those are always there. Absolutely. In the very first study, we did not find an association that survived multiple comparison correction. If you look at that paper, we looked at 36 areas times 2 hemispheres, so it was a very high number. And so the ones that I showed are the strongest of all. The sensory motor cortex, the primary sensory motor cortex, and the supplementary motor area do show up. Yeah, absolutely.
They fall out as you control for.
No, but again, we're looking at each one of them in isolation. Right now, we have looked at individual region with the outcome. You can't put all these regions in the same model because they're highly correlated with each other. So they are there, and it's a good, clearly reinforcing finding that we are looking at what we should be looking. Yeah, question?
Did you do anything sort of simple like looking at carotid blood flow and looking for plaques in the carotid and seeing whether there was a change in cerebral blood flow at that level, or even more sophisticated with MRI, cerebral blood flow?
No, we do have cerebral blood flow data that we just collected, and we haven't looked at those associations yet.
And sometimes in this situation, if you can, I realize with patients it's much more difficult. This is where animals are so valuable. If you do a longitudinal analysis, you can see which factor changes first, which is perhaps a little indication of causality. So, if I had the ultimate control over these animals, sorry, these patients,
They're not even patients, actually. They're individuals living in the community.
One would try to study them perhaps at 6-monthly intervals when they got to that age of 55 or so and see whether, I would put, as I said before, quite a bit of money on blood flow. I just would, as a physiologist.
Absolutely.
And so it would be very interesting to see if the patients who succumb to a slower gait speed earlier have a sort of preceding antecedent change in blood flow.
Yes, no, absolutely. I can see where you're going, and I think that's one of the things that we were trying to do when we obtained repeated brain MRIs. And as we were obtaining the same, we were documenting their cardiovascular health and physical health and so forth. We discovered, though, that, we just started looking at the follow-up MRI, brain MRI, but we discovered that 5 years when you are, I think they were already in their late 70s when they had the first MRIs. So they were in the mid 80s when they had the second MRIs. It's an extremely long period of time. And I wish we had had more frequent brain MRIs to capture changes, as those changes were actually happening. The other thing is that inevitably you have a survivor bias. That I don't know how you address it. One way to address it would be to have everybody donate their brain so that you have a chance of looking at their brain, and that Dave Bennett's studies in Chicago are very helpful in that. But then there is also a huge problem because they die whenever they want to die, and you have an interval of time that varies between the last time you saw them and the time when you have the brain. So I agree that animal models may be one way to study the evolution of these changes and how they begin. I too believe in the vascular hypothesis of brain changes, and that's why a lot of what we have done has to do with cardiovascular factors. We looked at arterial stiffness and diabetes and systolic blood pressure. Only one thing, that's why I think vascular factors play a role, because of the watershed distribution, and the regions that we see are most strongly related to slow gait, which are partially overlapping with the regions that are strongly associated with information processing, are also most vulnerable to lower oxygenation and because of their watershed vascularization. Yeah.
What about, approaching it from a different way, what about folk who had traumatic brain injury?
What about that?
Is that one way you could look at the cardiovascular problems? You expect?
I'm not sure about that, actually. The problem with traumatic brain injury is that the nature of the damage is really wild.
Traumatic brain injury. So you would expect those people to have problems with their blood flow.
I'm not sure about. I'm not sure. I know what I see that.
But do they have changes in gait?
Excuse me?
Do they have changes in their gait? Maybe they're accelerated models.
So they do gait speed. One of the things that we're trying to do with this cooperative agreement is to convince every investigator in the world they should add gait speed measures. If you're here listening, please do it to their studies. TBI patients do a lot of things that have to do with upper body, a lot of test hand dexterity. But I know at the very least a big study of TBI that we have in Pittsburgh that do not have gait speed measures. Yes?
I think gait speed is fantastic. We started doing it in some of our older baboons. But I'd like your take on this. If you look historically in the human as well, the phenotype is changing. We're in an epidemic of obesity.
Mm-hmm.
And that per se, I would have thought, I don't know, so this is my question, is going to change gait speed by itself from a physical point of view. So has anybody got any historical data so they can relate it to any changes like that?
That's interesting. So a lot of what you're saying in epi terms is a cohort effect, birth cohort effects, life course approaches. That is exactly what we need for the study of aging. Studying the last 10 years from age 70 to 85 until when a person dies means just looking at the result of processes that have been at play for decades, ever since we were born. We are engineered to age the moment we're born, right? So I think it would be fascinating and interesting. It's absolutely what is needed. The new GSA, the upcoming GSA conference, does focus specifically on aging across the lifespan. And my hope is that we will see a lot more of studies that have to do with earlier age than 65 and older, which is the typical cutoff.
Yes, I love that idea, but I was also thinking of not aging in the same person.
Yeah, the birth cohort effect, absolutely. Yeah, that's what you're saying, birth cohort effect, absolutely. That would be really interesting.
Like I said, they'll all have heavier BMIs now than they had 20 years ago, without any doubt. The BMI of an average cohort of any human population will be higher now than it was 20 years ago in any country in the world.
There have been a lot of changes in the way we manage blood pressure as well, right? And the number of other factors, right?
Yeah.
So this is, yes.
Just a comment. You said one of the earlier slides that you had corrected
BMI is associated with gait speed. That's what you're asking? Yeah. And that's why he's saying, would it be interesting to say the size of the effect of obesity across birth cohorts? That's a very interesting. So the studies that I know that have examined birth cohorts effects have focused on dementia, not on gait speed. Because, again, the gait speed is not yet an outcome in aging that is being included in epidemiological studies. Not yet.
So much cheaper than the MRI.
Thank you. I mean, when are you going to be a reviewer of my grants? All right, so I wanted just to show you some of the critical papers here that indicate what are the factors that are most related to gait speed, because that was kind of offered me sort of a roadmap to see what risk factors should I be looking for when I looked at the brain areas most related to mobility that could be affected by these factors. And so you have MI and stroke, and you have dyspnea as well here. This is one of the pioneer papers by Guralnik. And here you have a long list of factors. Tell me if there's something missing, but just about anything that happens with older age is related to slower gait, is related to a number of other things. That's what I find challenging and intriguing of studying the process of aging, and I'm sure you all do, being at the Barshop Institute, and that is a very complicated matter. It's a multifactorial problem. There are evidence, again, that lipid lowering could be related to better motor performance measured using the short performance, I think it should have been SPPB, short physical performance battery. And then blood pressure lower regimen also reduces the disability risk, and possibly through lowering the risk of stroke and vascular events. And then we also found a direct association between high blood pressure and gait slowing over 18 years of follow-up. There was no way, by the way, that could explain this association. I put just about anything in the model, including the brain MRIs. I failed to explain the association between blood pressure and gait speed. What is the pathway? What else could have been? Ideas, yeah.
No, but I think I don't heard it, but I was thinking about the vascular, and maybe this is my ignorance by MRI and what is exactly that you're measuring. You were told us that you measure integrity of those networks, and I guess that that's by activation. Essentially. So is it fair to say that you're actually measuring in a certain sense? I mean, it's obviously the whole system together, but you are actually measuring the ability also of those microvessels to provide the signal for your MRI. So basically, if you see, is that something that could be fair to say, or is it?
Not for the structure. Not for the structure. Not for the structure. For more sophisticated measures, which we have only begun looking at, like resting state or ASL, that's why I think that's very interesting, because it's primarily vascular, cerebral blood flow, like it was suggested earlier, yeah, but not so much for the structure. So what we said in the paper is that the reason why we couldn't explain those associations, the association between high blood pressure and gait slowing using the brain MRIs, because those were those ancient brain MRIs that looked at the brain as a whole. We couldn't really identify spatial network. It was kind of, I don't know. It is one of the possible reasons, but I have to say that this analysis was done in, I don't remember how, I think the cohort was 2000, and I just could not believe that association was so strong. It just would not go away. It was a very strong association. And we coded hypertension and high blood pressure in a number of ways: untreated, treated, uncontrolled, controlled, and we used systolic blood pressure as a continuous and so forth. Anyways, years later, about probably almost after 10 years, I don't remember when we published this, but years later, we looked at another group of older adults. This time, I looked at blood pressure as a continuous. It was measured for the first time in 97, 98, and then at regular intervals until 2007-08. That is when we did brain MRIs. So we could identify 5 groups. This is a clustering analysis just based on data. Okay, you feed the algorithm your data, and it creates these 5 categories. First interesting thing is that those who were stable, say started low 10 years ago and stayed low, started at the moderate level and stay moderate. Those who were high and stay high, this represented almost 80% of all the cohorts. When you get people who were 70, 69 years old and older, I think they remain stable. Most of them don't change much. I think that's something very important to consider. They remain stable. You're not going to see a lot of variability. So that's important too when you consider, so how many people do I need if I want to study variability of these measures? We were still able to detect two patterns of variability, those who were high and then decline and remain moderate for a substantial amount of time, and those who were really pretty variable all throughout, okay, and remain pretty high. And these two groups were those who were most likely to have hyperintensities in the white matter, which is a marker of small vessel disease, cerebral the small vessel disease. And so we thought that that meant that not only being high is important, but how variable you are over time is important for the integrity of the brain. So maybe reducing variability more, or in addition to reducing the absolute level, could be a strategy for improving those brain networks and possibly improving gait. Let me just skip through that because I think the discussion has been very rich, and I think if you can read the papers. What I'll tell you here is the same of what we wrote. The arterial stiffness related with brain integrity, specifically in the frontal lobes, diabetes related with integrity of certain regions. This is very important. This is physical activity and fitness in adults older than 79 are related to higher microstructure integrity, again of the frontal areas, but also of the temporal areas. This mid-temporal lobe comes up a lot, mostly the cingulate cortex, perhaps even more strongly so than the hippocampus in this group. So physical activity is one of the things that I've become very interested in, and seeing whether modification of lifestyle, increasing physical activity, could be related to improvement in the brain. We did this study 2 years ago where we saw that actually participating to a physical activity program that required also walking 3 times a week, this was the life pilot, was related to higher levels of activation when performing, again, the digit symbol substitution task, greater accuracy, and being faster at this task. This was 2 years after they had actually the study had closed. So this study indicated that if you take people who are sedentary, so this was a similar level of kilocalories per week, that's a sedentary lifestyle. If you take individuals who have been sedentary for most of their lives, you have them exercise for a year. Again, it's not an active exercise. They're not really waiting, in a gym. They're walking 3 times a week. If you have them do that for a year, and then you let them go, they go back to their homes, they don't come to the study anymore, you call them back 2 years later, there is not only the PA group, the group who had been in the exercise, has kept somehow the new lifestyle, but this group also had a higher, better patterns of activation, meaning higher levels of brain activation, higher accuracy, and lower reaction time. I got very excited. Of course, there is a huge selection bias here, right? Maybe those who came back did have a higher activation to begin with. I did not have an MRI here to prove it, but now we do, and so we should be able to distinguish to what extent having higher levels of brain activation could predict those who are more likely to adhere to a program, those who are more likely to benefit to a physical activity intervention program. So I think you have asked all of these questions here, and there is evidence the prefrontal cortex and information processing speed are modifiable. And this evidence comes from intervention studies that have used lifestyle and physical activity intervention, cognitive training, that's the question that Veronica was pointing to, even pharmacological interventions to improve information processing speed. These are studies done primarily, however, in Parkinson's disease patients from Jeff Hausdorff in Israel. So this is really what we need to go to provide a proof of concept, right? That there is a causal relationship. Is the modification of these risk factors improving mobility, and does it go through the central nervous system? I think that's pretty much all. I wanted to show you some of the people who work or have worked in our lab. You have seen their names. Some of us are now transitioning toward disease models of accelerated brain aging, like type 1 diabetes. And what we have been doing in the past few years, really in the past 4 or 5 years, has been to try to study the small vessels in much greater detail using the 7 Tesla neuroimaging, because I do believe that it's definitely vasculature plays a huge role. And so we are studying that. We're doing tracing of the small vasculature. We are segmenting the hippocampus to isolate the denta gyrus, where most of the neurogenesis occurs. And maybe that is, when I say the brain is important and has plasticity, I haven't really told you one measure of brain plasticity. You know, I told you less small vessel disease or less atrophy. But what if we were greater capacity to regenerate, to produce new neurons? And the only way to know that in vivo is to have a neuroimaging technique that is strong enough. And that 7 Tesla gives you this opportunity. These are studies of resting state at the fault node network and DTI measurements. So I hope maybe some of you can come and visit and see some of the things we're doing. And I hope we'll have some new interesting results pretty soon. Thank you.